Hello, everyone, and welcome to today's webinar, Deciphering the Crosstalk between Immunosuppressive Cells in Pancreatic Cancer using 3D Light Sheet Microscopy. I am Serena Demers of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Miltenyi Biotech. To learn more, visit miltenyibiotech.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well, if you have any trouble seeing or hearing the presentation. I'd like to now welcome our speaker, Dr. Carole Siret, light sheet microscopy expert at Centre de Immunologie, Marseille Luminy. Carole, you may now begin your presentation.
Hi everyone, thanks for attending this webinar and thanks to Miltenyi for giving me the opportunity to present my work on the crosstalk between myeloid-derived suppressor cells and regulatory T cells in pancreatic cancer. Pancreatic ductal adenocarcinoma, also called PDAC, is the most common malignancy of the pancreas. It's a highly aggressive disease with a 5-year survival rate lower than 10%, projected to be the second leading cause of cancer-related death in 2030, and with 4 to 6 months median survival after diagnosis. The main reason for this poor prognosis is the resistance to current therapies. This therapeutic failure is due to a low level of immunogenicity of neoplastic cells and to a robust immunosuppressive tumor microenvironment. This microenvironment is a real barrier to cytotoxic agents and limits effector T cell infiltration. This suggests that this tumor microenvironment could support cancer initiation, progression, and the development of metastasis. This microenvironment is highly fibrotic and composed of an extensive infiltration of immunosuppressive cells such as tumor-associated macrophages, myeloid-derived suppressor cells, and regulatory T cells. In this study, we have been particularly interested in MDSC and Treg cells, which represent a nation's essential class of immunoregulatory cells in PDAC. MDSC are key regulators of immune responses in many physiopathological conditions, including cancer. PDAC cells secrete factors involved in the induction, recruitment, and survival of MDSC, leading to MDSC accumulation. And this accumulation in peripheral circulation of patients has been related to the extent of the disease and associated also with a poor prognosis. Concerning Tregs, they are crucial for immunohomeostasis and peripheral tolerance. In PDAC, Tregs recruitment occurs early and are present in pre-malignant lesions. Their prevalence increases with tumor progression, and their depletion slows tumor growth and prolongs survival. Although a strong influx in MDSC and Tregs has been described in PDAC, there is no evidence on the presence of the interaction between these cells. And it's also unclear if Tregs can act on MDSC and shape their functional differentiation. That's why we wanted to decipher the crosstalk between MDSC and Tregs. For that, we used an orthotopic tumor mouse model. We injected PANCO2 cells, a mouse pancreatic cancer cell line, and matrigel directly in the pancreas of the mice. We waited 3 weeks to harvest the tumors and analyze them by cytometry, confocal microscopy, and light sheet microscopy. First of all, we wanted to characterize the immune populations present in the tumor. We did immunochemistry 3 weeks post-inoculation and used an anti-GI1 to stain MDSC, anti-FOXP3 for the Tregs, and anti-CD4 and CD8 for effector T cells. We observed a strong influx of immune cells characterized by an infiltration of MDSC and Tregs in the tumoral pancreas. No immune cells were detected in a normal pancreas. We analyzed also the tumor biocytometry and showed in a tumor-bearing mice a huge increase of CD45 plus cells. In this population, we show the presence of MDSC population, stained by CD1, CD11B, and GR1. We characterize phenotypically this population. The granulocytic population, LY6G plus and LY6C low, and the monocytic population, LY6G neg and LY6C high. We could observe a strong expression of B7H1 and CD40 on both subsets derived from tumors. However, the upregulation of F480 and CD124 was more substantial on the monocytic subpopulation. We also showed the presence of Tregs in this CD45 plus population. We observe around 6% of CD4 plus cells, and in this CD4 plus cells population, alpha are Treg, so CD4 plus and FOXP3 plus. In fact, currently the most commonly used marker for Tregs identification and characterization are CD4, FOXP3, CD25 for the interleukin-2 receptor, and the CD127 for the interleukin-7 receptor alpha. We demonstrated that most of the CD45 plus CD4 plus FOXP3 plus are CD25 high and CD127 low. We show also the immunosuppressive function of that via the TGF-beta secretion and the expression also of the markers CTLA4. The tumoral Tregs upregulated the TGF-alpha and CTLA4 expression compared to the splenic counterparts. We also examined the expression of CCR5, chemokine receptor, so at the surface of the Tregs. In fact, this receptor could mediate the traffic of Tregs to PDAC tumors. MDSC and other cells in the tumor microenvironment, like pancreas stellar cells, can secrete at high level the CCL5. So here we demonstrate that 20% of our tumoral Tregs express CCR5 at the surface, and we study also the expression of the integrin alpha E, the CD103, because in fact this integrin has been reported to be an alpha mark of a tumor infiltrating Tregs in mouse colon cancer. So this Treg, this CD103 positive Treg were described as potent suppressor of an anti-tumor immune response in this cancer, in colon cancer. And in our model, we show that more than 70% of these Tregs express this molecule. We analyze after the recruitment and the activation of the CD4 and CD8 T cells. So a low infiltration level was observed in a tumor, around 3% for CD8 plus cells and 4% for CD4 plus cells. We assess the activation state of these cells and show that T cells downregulate CD62 ligand and upregulate CD69. More than 88% of the intratumoral CD4 and CD8 from PDAC strongly downregulate the CD62 ligand, and the expression of CD69 were about 24% for CD4 and 33% for CD8. Since these phenotypic markers cannot conclusively determine if infiltrating T cells are truly naive or functionally inactive, we measure also the intracellular level of the effector molecule interferon gamma, and our data show that 10% of the CD4 and 18% of the CD8 T cell from tumor produce interferon gamma, suggesting an activated phenotype. So PDAC is characterized by a strong accumulation of immunosuppressive cell population, MDSC and Tregs, associated with low levels of activated CD4 plus and CD8 plus cells. So now we wanted to characterize the functions of these cells. To assess the suppressive potential of MDSC, we co-culture MDSC with CD8 plus cells and show by cytometry that CD8 T cell proliferation is suppressed by tumoral MDSC in dose-dependent manner. Indeed, when we increase the ratio MDSC cells on CD8 T cells, we increase the ability of MDSC to suppress the T cell proliferation. We also observed that when we co-culture stimulated the CD8 T cells with MDSC from tumors that the tumoral MDSC can induce the death of CD8 T cells ex vivo. So CD8 T cells were co-cultured with MDSC and stained with cell death markers such as 7-ALD and annexin 5. And we observed that the percentage of alive cells decreased when the ratio MDSC from the ratio MDSC T cells increased. So next, we examine the presence of MDSC functional molecules such as ROS for reactive oxygen species and arginase 1, which are essential for the immunosuppressive capacity of these cells. We observe an important upregulation of this molecule by MDSC from PDAC compared to CD8 T cells and their splenic counterparts. After we investigated the suppressive function of Tregs, so target cells, so CD4 plus CD25 neg, were cultured with Tregs, CD4 plus and CD25 plus. And we saw by cytometry that target cell proliferation is suppressed by tumoral Tregs. So MDSC and Tregs from pancreatic tumor are able to suppress effector responsive in PDAC mice. So after we wanted to know if this cell MDSC and Treg could interact physically, indeed, the interaction between these cells in different cancer models have been proposed to play a role in shaping the tumoral immunosuppressive environment. But until now, there is no evidence that this crossover could involve direct interaction. So first, we determine by confocal microscopy the prevalence in pancreatic tumor, the prevalence of MDSC, so here in green, and the prevalence of a Treg here in red. And here also the nuclei are stained in blue. So we observe that the Tregs are located in close contact to MDSC in pancreatic tumor, and neither MDSC nor Tregs were observed in the naive pancreas. So to have a 3D view of this tumor, we decided to use the lighted microscopy to further study this interaction. So with a lighted microscopy, you can image a whole sample and do 3D reconstruction on numerous software, for example. So in this experiment, we stain PDAC tumor, so with the same antibody as used in confocal microscopy, GR1 for MDSC in blue, FOXP3 for Tregs in green, and we also stain the blood vessel with an anti-SMA in green. The tumor, the stain were cleared, and after image on a lighted microscope, the ultra microscope 2 from Milton Biotech. And the purpose of this experiment was to have an idea of the cell repartition of MDSC and Treg in the tumor, and see if we can identify also interaction between these cells. So we can observe, so here on this snapshot done so with Imaris, that MDSC in blue and Tregs in red were observed so in close contact as in confocal microscopy. So with Imaris, we can also generate 3D video, and here you can see on this video like before. So it's a little bit fast, but you can see like before the blood vasculature in green, the Tregs in red, the MDSC in blue, and also in yellow, here for example, the Tregs in interaction with MDSC. So we also identified interaction between these cells with a lighted microscopy. We quantify this interaction with Imaris. So indeed, with this software, we can calculate the distance between two different populations and see if this cell could be in interaction. So we quantify this interaction in 3 different tumors at 3 weeks after inoculation, and identified around 20 interaction per tumors. But these events are rare, and this probably due to the high dynamics. We also quantify this interaction at different points of the tumor development, 1, 2, 3, and 4 weeks, and we observe the peak of interaction at 3 weeks and after the number of interactions decrease. We confirm also the presence of this interaction by video microscopy. So our data demonstrate physical interaction between Tregs and MDSC in PDAC. So after, we wanted to evaluate the influence of MDSC on the accumulation of Tregs in vivo. For that, we depleted MDSC cells in PDAC mice, and we injected intraperitoneally an antibody, the RB6, in the mice, and it results in a near complete elimination of MDSC in spleen, but also in pancreas. And we also observe an inhibition of the Tregs recruitment in pancreatic tumor. So the systemic depletion of MDSC can inhibit the recruitment of the Tregs in a pancreatic tumor. To further investigate the cellular mechanism of MDSC and Treg interaction, we assess the ability of MDSC to promote de novo development, expansion, and recruitment of Tregs, and for that we co-culture Tregs and MDSC from a tumor. And when we co-culture these cells, we show an increase of the Tregs population in presence of MDSCs. And if you put a transwell chamber to separate the two populations, the number of Tregs decrease. So this suggests that Tregs induction by MDSC is due to cell-cell interaction. And vice versa, we tested the Tregs impact on MDSC. So we did a similar co-culture, and we assessed the MDSC viability. So after 24 hours and 48 hours of co-culture, the percentage of alive cells, so alive MDSC, strongly increased in the presence of Tregs isolated from tumors. So our finding revealed that Tregs can also affect the survival and or the proliferation of a tumoral MDSC. MDSC are able to induce Treg T cell proliferation and or development in a cell-cell dependent manner. And we also show that Tregs cells can affect the survival and or the proliferation of MDSC. So to finish, we wanted to determine if MDSC and Treg could be in interaction in the human pancreatic cancer. So for that, we study human PDAC. So we had access to parafina embedded tissue section for human PDAC, and we show, so on this section, the presence of T cell, CD4 and CD8 T cell, and we also stained MDSC here with an anti-11B and an anti-CD11B and an anti-CD15, which are the markers for human MDSC. And it's not shown, but no immune cells were observed in normal pancreas. And with a polyline kit, we also wanted to determine if we can see interaction, Treg and MDSC cell interaction. And we observe this interaction here in more than 50% of analyzed human PDAC. So here the MDSC are in pink and Tregs are in blue. So to conclude, PDAC is characterized by a strong accumulation of immunosuppressive cell population, MDSC and Tregs, associated with low levels of activating effector T cells. For the first time, in a tumoral context, we identified by light microscopy MDSC Tregs interaction, which occur via direct physical contact. This interplay add of biological relevances, the in vivo depletion of MDSC led to a significant reduction of Tregs in PDAC. And a new aspect also of our study was MDSC Tregs interplay's contribution to the establishment and or development of the immunosuppressive microenvironment in PDAC. Indeed, we showed that MDSC are able to induce Tregs in cell-dependent manner, and also that Tregs can affect the survival and or the proliferation of MDSC. But at present, molecular partners involved in this interplay are not known, and further studies are needed to investigate this pathway. Thank you for your attention, and thanks to Milton.
Thank you, Carole, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. Let's get started. Our first question is, do you have any candidates for the molecular partners implicated in the MDSC Treg interactions?
So, yes, one candidate to consider could be first the B7H1, PDAC1, because in mouse melanoma, for example, Tregs can shape the functional differentiation of MDSC melanomas, and Tregs can stimulate this molecule expressed as MDSC. But also CD40 and CD80 molecules could be also interesting to investigate. In fact, these co-stimulatory molecules are also present on MDC in mouse colon cancer and also in ovarian models. And their presence are also associated with Tregs accumulation. So these candidates could be.
Great, thank you. The second question, did you test if any other cells besides Treg could interact with MDSC?
So yes, we only tested, in fact, gamma delta T, but we did not observe any interaction. Because in fact, these cells, it was shown, in fact, in hepatocarcinoma that gamma delta T cell can exercise the pro-tumoral function by recruiting MDSC, but this interaction seems to be indirect. And yes, but in PDAC, in the literature, there is other interaction described, like Tregs with CD11C DC, and also other interaction like Tregs and TAM for tumor-associated macrophages. But we only tested gamma delta cells.
Thank you. The third question, is this crosstalk between MDSC and Treg present in other cancer types?
Yes. In melanoma, yes. In melanoma, via the same molecule B7H1, yes, MDSC expansion and their recruitment occurs in a Treg-dependent manner, but we also observe this crosstalk in lung cancer, if I remember well, also in colorectal cancer and breast cancer.
Great. And what is your protocol to clear tumor samples?
So our protocol to clear the tumor is based on BIBB protocol. So this method, in fact, is based on dehydration and delipidation by organic solvent. BABB is a mix of benzyl alcohol and benzyl benzoate. And after dehydration and delipidation, there is a step of aerial matching. And because, in fact, most of the biological samples are opaque, and we need transparent sample to use light kit. And so we have to remove water, lipids, and so on.
Fantastic. And another question we have here, how exactly do you determine MDSC Treg interactions using light sheet microscopy?
So we determine this interaction by Imaris software. In fact, with Imaris, you can count the cells, the stained cells, in the different fluorescent channels with this spot function of Imaris. And when you have determined this number of the population, you can use also a MATLAB function in Imaris, which allow you to measure, in fact, the distance between two cells from the centroid. And in our study, the lymphocyte, we estimate that the lymphocytes are around 10 micrometer in diameter, and for the MDSC 20 to 30 micrometer. So we ask to Imaris to determine the number of the interaction when two cells, in fact, are at the distance equal or less than 20 micrometer.
Awesome, thank you. And we have a question coming in from Marcus. What is the basic principle of light sheet microscopy?
So light sheet microscopy, in fact, you have to clear your sample. You can work on a whole sample. So, and the principle of the microscope, and you have a sheet of light, and you illuminate your sample with this sheet. And in fact, this sheet of light is orthogonal to the objective, and you can acquire so only the plane illuminated by the light sheet. And when you have a big sample, you determine the stack that you want to acquire, and after the acquisition, you have a stack of picture, and you can reconstruct with this stack of picture your entire sample.
Thank you. And we have a question coming in from Ralph. How did you isolate the MDSCs and Tregs?
So for the isolation of the cells, so we harvest the tumor 3 weeks after the inoculation. And so we dissect them in a little fragment with scissors. And we did the incubation with collagenase type 1. If I remember well, it's 30 minutes at 37 degrees. And after the suspension is filtered, and yes, we sorted the same.
Thank you. Our next question is coming in from Alejandro Lopez. It's a two-part question, and he's asking, what precautions should be considered to image a tumor in light sheet? And do you suggest a protocol for clearing and imaging in light sheet?
Sorry, can you repeat the question?
I can repeat the question. What precautions should be considered to image a tumor in light sheet? And do you suggest a protocol for clearing and imaging in light sheet?
For tumors, so yeah, so we use a protocol based on BABB, but you can use also, I think, IDISCO protocol. We didn't use IDISCO protocol because some of immune markers are very sensible to methanol, and in the IDISCO protocol, we have a pre-treatment with methanol. So that's why we didn't use this protocol. But I think for tumors, iDisco will be the best, because we have this pre-treatment with methanol that can increase the permeabilization of the tumor and so increase the penetration of antibodies inside the tumor. We have also a step of bleaching, and this step of bleaching can be very usable when you have autofluorescence, and in tumors sometimes you have a lot of autofluorescence.
Great, thank you.
Yeah,
our next question is coming from. Oh, go ahead. I'm sorry.
Yeah, just to mention that when you use iDisco protocol, you have to test your antibody before, because as I said, you can have problem with methanol. So you have to test your antibody with on section pretreated with methanol before, to be sure that your antibody can work with this protocol.
Great, thank you. Our next question is coming in from Matthias. Is the phenotype a general occurrence in both human and mouse tumors, or is it rather a localized, in quotations, either periphery or core of the tumor?
So in fact, in human, we identify interaction on some tumors on paraffin section. Localized. But in fact, with a human, we had access only on a very small piece of tumor. So for the localization is difficult to assess, because it is very small, and we don't know exactly where we are in the tumor.
Great.
But
thank you.
Yeah. Okay.
Our next question. Are you ready for our next question?
Yes.
Our next question is coming in from Virginia. It's a two-part question. Can you repeat which antibody you used for MDSC's depletion?
For MDSC depletion.
SC removal.
Excuse me, I didn't hear you well. What? Can you?
The first part of this two-part question is from Virginia. Can you repeat which antibody you used for MDS depletion? And so the second part of the question?
Yeah. So for the antibody is the RB6-8C5 antibody. It's an anti-GR1. Do you hear me?
Yes, I'm so sorry. Did you hear the first part of the question?
I don't hear you. Hello? There is somebody?
Hello. Did you hear the first part of the question from Virginia?
So for the antibody, I hear. So the antibody is the RB6-8C5 antibody. It's an anti-GR1 antibody.
Fantastic. Thank you. And the second part of the question, did you study the effect of MDSC removal and subsequent reduction of Treg on the cancer cell population?
No, not on the cancer cell population. No.
Okay, thank you so much. Then the next question we have coming in is from Paul. Do you have evidence for a role of CAFs in facilitating or inhibiting the interactions that you described for the immune cells?
No, we didn't assess this question, but yes, it's known that CAF can have interaction with MDSC and Tregs, but yeah, we didn't assess this question.
Thank you. And our next question is from Matthias. It says, but you are measuring nucleus distance to cytoplasm, so you can never get sure of direct interaction.
No, in fact, Imaris do the measure from the centroid of the cell, so it's the nucleus to nucleus. So, and we hypothesize that lymphocytes are 10 micrometer in diameter, and the MDSC a little bit more, 20 and between 20 and 30, and we estimate that 20 is a good distance to be sure that the two cells are in interaction.
Great, thank you. Thank you, Carole. Do you have any final comments for our audience?
No.
Thank you, Carole. Do you have any final comments for our audience today? Hello, Carole. Thank you so much, Carole. Do you have any final comments for today?
Yes, thank you for your attention, and thank you for the question.
Fantastic. Thank you again to our speakers, Carole Siret, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Miltenyi Biotech, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today, and those submitted during our on-demand period, will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available to replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye and thank you.